Today I'm going to be explaining the moss life cycle. So first I like to start off with deciding what stage the cell is going to be in. And an easy way to do this is to just draw a line straight down the middle in between the blue and the pinkish brown area and through meiosis and fertilization, and then label up here an N and then A2N and do the same on the other side. And the N basically represents haploid and the 2N represents diploid. And so you'll always know that the upper blue part by this line will always be in haploid cells and the bottom will be diploid. And the only way to get in between the two is from haploid to diploid is through fertilization, and then from diploid to haploid is through meiosis. So this is a cycle that'll keep going, but in terms of the chart, we'll start off with number 1. This is meiosis. It starts off the sporangium after fertilization, which is the last part of this chart. So we'll get to that a little bit later, but it grows into a spore, which is kind of the starting stage of it. And this turns into mitosis, which is basically the stage where it splits off right there, and it says that no two cells can be the same, indicating the gametophyte and the gamma-4. Some of the spores will continue on to becoming more archegonia because they have an egg inside of them, or some of them will become the male part, which is the antheridia. This leads us to stage 3. This is when the moss first grows on the spore as a protonema, which develops into a gametophore or a gametophyte. The photosynthetic part that you can see is the gamma-phyte. This is the male part, and the male part does the photosynthesis because it is the dominant part of the life cycle. The male part is going on to produce sperm by mitosis, very dependent on water, which is indicated by the raindrop because it's given by rain most of the time. They have phalangated sperm, which means the sperm has to swim into the egg and they have phalangium on them. Then they can fertilize the egg. They swim up and then down. For fertilization, the sperm and the egg are both needed, which are both haploid cells, coming together to form a zygospell, which is the brown organism right in here. And once it is fertilized, it'll hold the zygote and it'll turn into an embryo, which will divide because of mitosis, and the zygote will turn into the young sporophyte. The brown balls at the end are going to produce spores. So while looking at the mature sporophytes, this picture on the next slide really is a picture of that basically. And these things at the ends are the sporophytes, and then the bottom parts are the gametophytes. And that's basically what the sporophyte grows off of. So we're going to start by looking at the differences between monocot and dicot stem cells. So the main difference is going to be in the vascular bundles. In monocot cells, the vascular bundles are scattered everywhere with a specific arrangement, as you can see right here. And then the dicot stems vascular bundles are arranged in 1 to 2 broken rims that follow a definite pattern. So that's right here. And there is no bundle sheath actually on the dicot stems, while in monocot stems, the bundle sheath surrounds the vascular bundles, so they're all around them. The hypodermis is made up of sclerenchyma in monocots, while the dicot cells are made up of the parenchyma. And in the hypodermis, it secretes the Chintus cuticle, while in the dicot cell and in the monocot cell, it does not do either of these. The vascular system is made up of cortex and stele, and these are the two distinguishing regions in the dicot cell, while the vascular bundles in monocot cells are scattered and there is no specific cortex or stele found in them. The cortex is beneath the epidermis, and this is basically to transport materials to the center cylinder of the root. And this can be used for food storage as like starch. The pith is in the center of the cortex in dicot stem cells, and it basically holds the starch in the water for the intercellular space. And in the middle of the vascular bundles is the pith, and basically the vascular bundles do not keep reproducing over life, they just keep growing, and inside the vascular bundles is the xylem and the phloem. The phloem is responsible for the production of carbohydrates in the cell, and it also moves the sugars and organic compounds of the plant from the leaves down to the stem. The cambium is responsible for the secondary thickening and separating the phloem from the xylem. The xylem is basically, the responsibility of this is to strengthen and support the stem, and it transports the water from the roots to the leaves. Monocot stems are much less complex than the dicot stem, but these also do have a complex vascular bundle, and basically these are all scattered around the outside edge, and there is no specific pattern to them. The epidermis in the monocot cell still has the same responsibility as the dicot. It's there still for the protection of the plant and to keep the water in, making sure there's not too much water loss. And the last main understanding to monocot stems is that the ground tissue is called parenchyma, so when the two are used intermixed with each other, they mean the same thing.